What is a plasmid? Plasmids are fragments of double-strand DNA that can replicate independently of chromosomal DNA and usually carry genes. Characteristics: a small size allowing circular DNA is more stable when removed. Self-replicated: you can make an endless number of copies of plasmid by growing the plasmid in bacteria. Stable: plasmids are stable long-term either as purified DNA or within bacteria. Functionally in many species and can useful for a diverse set of applications. Plasmid can drive gene expression in the wide variety of organisms. Vector elements. These elements allow for the propagation of the plasmid within bacteria. The restriction enzyme site allows for the cloning of a fragment of DNA to be substituted into the plasmid. Origin of replication. Antibiotic-resistant gene. Multiple cloned sites insert promote region, selective market, and primer binding site. Types of plasmid: cloning plasmid, expression plasmid, gene knockdown plasmid, reported plasmid, and viral plasmid. There are many different characteristics that a microorganism can attain when acquiring a plasmid. One of the most common characteristics is resistance to a certain antibiotic like ampicillin. Provide microorganisms with the ability of creating symbiosis. New plasmid can allow the microorganism to change color in presence of some reagents like UV light. Plasmid can also provide the microorganism with virulence, therefore a not virulent microorganism can become virulent. Microorganisms could also be responsible of making microorganisms available to survive in presence of alternative and non-common carbon sources. Plasmid could be high copy and low copy, which allows the microorganism to have the characteristic of expressing a high or a low quantity of determined genes. A microorganism can also become resistant to heavy metals after new plasmid acquisition. There are plasmids that give microorganisms the characteristic of forming tumors in plants. They too can again have certain characteristics that allow the same to colonize certain tissues and host organisms. A microorganism can also acquire the resistance to a certain bacteriophage by getting a new plasmid. Plasmid extraction. Plasmid extraction involves the following steps: growing of the bacteria culture with a selection agent, collection of the leases of the bacteria, and plasmid purification. Difference in plasmid extraction and nucleic acids. The difference between the methods of nucleic acid extraction and the plasmid is the portion of cell lysis. It seeks to create small pores in the cell membrane so that only the plasmid can live and the chromosomal DNA cannot live. Another difference is the neutralization. Chromosomal DNA to interact with potassium and precipitate is achieved from only remove the plasmid. And the last one is the time to renature of chromosomal DNA. Alkaline lysis. It is based on the properties between the naturalization and renaturalization of plasmid DNA and chromosomal DNA. Alkalinization with sodium hydroxides in the presence of a detergent such as SDS causes cell lysis, the naturalization chromosomal DNA and protein, and lysis of plasmid. Neutralization of the medium in the presence of high concentration of salts, potassium acetate. This causes precipitation of proteins. This is because by draining with detergent and insoluble of the potassium salts and chromosomal DNA. The insoluble aggregates of protein and chromosomal DNA are removed by centrifugate and the plasmid DNA remained in the supernatant. Mini preps. They are extraction from a small volume like 1 to 1.5 milliliters bacterial culture which allowed recovery and reflected circular DNA on chromosomes DNA. Mini preps with a state buffer and lysosome use special detergents to open small pores in the cell membrane so you can leave only the plasmid. Plasmid and isoforms. When plasmid DNA is extracted from these bacteria, they can occur in 2 or 3 isoforms, supercoated, linear, and relaxed circular full length. So you can see 2 or 3 bands, and each band belongs to each isoform. The difference between the bands is because the volume occupied isoform is different, so they expand to migrate through the yield is different. The supercoated are more compact and therefore easier to migrate mind. Relaxed circular and linear migrate more slowly because they occupy a larger volume.
On figure 1 we can observe plasmid extraction and electrophoresis. It was carried out in a 1% agarose gel with 1x type buffer at 100 volts through 30 minutes and 120 volts through 15 minutes. The bands were visualized under ultraviolet transillumination. Lens 1 contains 1 kilobase layer oxygen, 5 microliters. Lane 2 contains contains plasmid sample from blue colony, 5 microliters plus loading buffer 4 microliters, and lane 3 contains plasmid sample from white colony, the same, 5 microliters plus loading buffer 4 microliters. On a more detailed observation, we can observe in orange sample degradation and possible contamination by salts. Also, we can observe in yellow circular plasmid. We can observe in green linear plasmid of a higher intensity, and we can observe in blue RNA contamination, which was supposed to be the supercoiled plasmid.
In prokaryotes, plasmid is double-stranded, circular, and is found in the cytoplasm. The cell membranes must be disrupted in order to release the plasmid in the extraction buffer. Glucose provides osmotic shock, leading to the disruption of the cell membrane. This is a buffering agent used to maintain a constant pH of 8. Plasmid can be protected from endogenous nucleases by chelating the magnesium ions using EDTA. Magnesium ion is considered as a necessary cofactor for most nucleases. Sodium hydroxide and SDS are used to disrupt the cell membrane, and sodium hydroxide also denatures the DNA into single strands. Acetic acid neutralizes the pH, and potassium acetate precipitates the chromosomal DNA proteins along with the cellular debris. Phenyl chloroform solution is used to denature and separate proteins from plasmid. Chloroform is also a protein denaturant which stabilizes the rather unstable boundary between an aqueous phase and a pure phenyl layer. The denatured proteins from a layer at the interface between the aqueous and the organic phases, which are removed by centrifugation. Once the plasmid DNA is released, it must be precipitated with alcohol. The plasmid DNA in the aqueous phase is precipitated with a cold ethanol or isopropanol. The precipitate is usually redissolved in buffer and treated with phenol or organic solvents to remove the last traces of protein, followed by reprecipitation with cold ethanol. Although E. coli produces mainly the most compact supercoiled plasmid DNA isoform, open circular, linear, and denatured isoforms are usually present due to conformational changes that occur within the bacterial host or during processing of biomass. It is well known that typically only one band, the circular closed form, is observed when a small amount of a plasmid sample is applied to an agarose gel. Standard agarose gel electrophoresis usually reveals two prominent bands, the circular closed form and another slower migrating band, commonly thought to be the open circular form. Overloading a gel results in a larger number of additional bands. According to results in Figure 1, 3 defined bands are shown. Nevertheless, two isoforms were distinguished. The first one corresponds to the linear and the second to an open circular isoform. Moreover, one can wrongly interpret the last band on both lanes as a supercoiled isoform, but it's clearly RNA contamination. The open circular, linear, and denatured topological isoforms are derived from the intact and undamaged supercoiled plasmid DNA by either single-strand nick open close or double-strand nick linear. Whatever the case, the resulting isoforms are likely to be less efficient in transferring gene expression when compared with the supercoiled isoform. This is especially true if the cleavage disrupts promoter or gene coding regions. However, purification of plasmid DNA isoforms by agarose gel electrophoresis is advantageous in terms of cost and availability. It is a slow process with low throughput. Furthermore, recovery of DNA from the gel is cumbersome and requires removal of the staining agent used for visualization. According to the international regulations, product quality is defined as the percentage of the supercoiled isoform relative to the total plasmid DNA. Accordingly, the purity of the supercoiled plasmid DNA must be 100% with a homogeneity higher than 97% of the supercoiled isoform from E. coli host. Therefore, according to the results, as the samples were not treated with RNAs, the supercoiled isoforms weren't isolated, and consequently the purity of the supercoiled isoform was null, and so did the product quality.
Conclusions. In order to obtain plasmids from transformed colonies, it was necessary to carry out a cell lysis, which in this experiment was alkaline lysis, the chosen method. There exist other methods similar to the one practiced, like isoprep and miniprep, which include other types of reactants like sucrose, other soaps, and fewer time. It is necessary to verify which are the goals and later choose the best method that better fits your necessities. Also, it is important to manage samples with care in order to have better results and follow the protocol step by step, so that the causes for variation and error get close to zero.